Okay. So today we're going to talk about one of the more interesting chapters in the book about adaptive laboratory evolution. So in terms of the previous material we've covered, this is focused on two issues. One is the fact that we can now study the objective function in the lab. And like I said, that's kind of the biologist's function. Don't find it in physics and chemistry and elsewhere. This is description of distal causation, or the process of adaptation. And we'll mention a little bit, again, the ability of genome scale models to predict phenotypic states and the optimal ones that we can then, in a lab, evolve a cell towards. Okay, so a few topics we're going to go through here. First, this is a new line of biological inquiry, namely that you can do controlled evolutions in a laboratory. You can repeat them. You can do them over and over again. You can do multiple replicates simultaneously, so you can get statistics on outcomes. We can use the phenotypic phase planes that we talked about before to interpret the outcomes, and these are calculated based on optimal growth rates. So if we have growth rate selection, these would apply. We'll talk about other selection pressures a little bit, but most of the lecture is on optimizing growth rates computationally and experimentally. Then we'll talk a little bit about determining the genetic basis. So this is something that has changed enormously since 2007. We can both sequence genomes now very cheaply and in mass to find any mutations or genetic changes that happen during adaptation. But we can also use the growing number of genome editing methods to introduce any mutations found in the endpoints back into the starting strain to assess causality of these mutations. I'll talk a little bit about the interpretation of the experimental outcomes, because that is not trivial. But there are many things we can do, some we cannot. And then we'll talk about some other types of evolutions. There are some natural evolutions we think we can mimic in the lab and build experimental models of. Some we can't. And then we'll talk a little bit about some general examples of complex adaptations. And in the end, I want to introduce the notion of a systemic perturbation variable. We talk about environmental perturbation by changing some environmental conditions. We talked about genetic perturbations by deleting a whole gene. Here we encounter a new type of perturbation, namely a causal mutation, which means if we push exactly there on the DNA sequence, the whole system goes from one state to another. If I move over a base pair or 2 or 3, or do a different mutation in the same spot, that may not happen. So these are very magical perturbations in the DNA sequence. Okay, so let's talk a little bit about ALE, or adaptive laboratory evolutions. So performing ALE, how is ALE performed? So there are different ways in which one can do this. So a couple of them are illustrated here. One is simply serial passage from one culture to the next. We take a small aliquot and pass it into a new media, let them grow, and you repeat this over and over again. You can do that in mid to late stationary, mid to late exponential growth phase to make sure the cells are exponentially growing as you pass them from one passage to the next, or one culture to the next. Or you can let them go into stationary phase in which the cells may mutate quite a bit in the stationary phase, and then you put them into a new culture broth, and then you'll have a lag phase and growth again. So both of those are done. And I think some of the classical experiments by Richard Lenski had the cultures go into stationary phase before passage. Many of the more recent examples that have been published do not do that. They pass in mid to late exponential phase. You can also do this in a chemostat over a long time. In a chemostat, of course, you fix the growth rate by the dilution rate. And it appears that the selection pressure there is more based on biomass yield rather than growth rate. So that's another way of doing it. Chemostats are a little more difficult to deal with operationally. You have to keep them stable over a long period of time. And very often you get wall growth. And if you have that kind of partitioning of your cell culture, that clearly introduces some problems. So there are ways to deal with that that we're not going to talk about here, so you can't do that. You can also do evolution on a plate, just pick a colony and pass it onto a new plate, streak it out, and then pick a colony and just pass based on colony properties, size or color or something like that. There's a number of evolutions now being done in a microfluidic device where you start with a single cell and look at it by passing it, or in a continuous way you can miniaturize the chemostats. You can also inject cells into an animal and watch the colonization process and look at the adaptations that happen as a species adapts to the host. And that will be an example of a real evolution. Or very close to real evolution. Of course, you start with a sterile animal in this case and you colonize it. We'll talk a little bit about that. And there are some evolutions that have been done based on electron acceptors or electron donors. And for electrosynthetic bacteria or bacteria put into fuel cells, you can actually alter the potential of the current, and you can be oscillating it or changing it, and you can force evolution in that setting as well. So there are many, many ways in which you can do ale. Now the one that does serial passaging in late exponential phase can be automated, and ale machines have been built, and here you can have many replicate cultures at the same time. In a sophisticated setup, you can do many simultaneous experiments, and the number of passages you can achieve per day will far outstrip what you can do manually. And also this is a very well controlled process. And the images to the right show the outcome of that kind of experiment. The top image shows your serial passaging, like a sawtooth curve where you pass at a fixed cell density, let it grow again. That little strip below that shows that that figure is just expanded from a very, very long set of experiments. You can see in the last few passages, for instance, the passage volume has been changed, and it starts at a slightly higher OD at the beginning. The middle panel shows many experiments being done simultaneously, and this looks like a mess. But you can computationally, as you go, determine the slope of that curve and determine the growth rate or the doubling time, and then plot that as a function of time. And that's what's shown in the bottom image there. So you see some of the growth rates are going up over time, whereas others are stable in that particular case. So this is nice. So you can get to scale here with replicates, different types of experiments, and so forth. Okay, so that's just doing ale. You can do it in many different ways, and you can automate some of these procedures. So let's talk a little bit about phenotypic phase planes and predicting optimal growth rates. Let's go through a little bit of history here. So shortly after the first genome scale models came out, this paper was published in 2001 that basically illustrates the procedure of predicting an optimal growth rate. You have a genome, go to the reconstruction, then you define the solution space, and then you optimize the growth function within that solution space, and you find that edge on the solution space that represents an optimal growth rate or an optimal yield, depending on how you do the calculations. Here is a specific example for E. coli growing on malate, and I think we looked at this before, where the phase plane is shown on top, which is the floor in that three-dimensional diagram. Line of optimality indicated in red, and you can see the points, experimental points, fall on that line. And in the three-dimensional rendering, you see the experimental points on the edge of that solution space. And there is this line of optimality which describes those points. In the top panel there's a red arrow that actually shows an evolution process. And in this case, it migrates along the line of optimality. Basically, the growth rates are increased under a constant substrate concentration, and the yield goes up, or the growth rate goes up. Okay, so now it turns out that the prediction of optimal growth rate isn't always accurate, and you would get a result like this. And this was the first data set of this sort that was published, and this is growth of the E. coli strain, this commonly used laboratory strain, on glycerol. And the points are not on that edge, predicted line of optimality. They look like they're on the surface of that polytope, but in actuality, they're inside the body of it. They are far away from the surface. And this is maybe an outcome you would expect when you're trying to grow a cell under conditions that it hasn't experienced before. You know, E. coli doesn't grow often on glycerol, and it may not know how to optimally utilize that substrate. So this was initially seen as a failure of prediction, but then when you started thinking about why did the experiment fail, was I missing something in metabolism? What could be wrong? But glycerol is only 3 or 4 steps away from glycolysis, and when you do these predictions on any substrate that goes into glycolysis, they work. So there's really, you don't expect that there's a lot unknown about glycerol uptake and metabolism in E. coli. So this hypothesis was then put forth that that pathway just wasn't optimal. It wasn't configured to support optimal growth of E. coli under those conditions. And that led to the sort of an evolutionary experiment in the lab that we talked about. And interestingly enough, these initial states over evolutionary time moved around in that polytope and eventually found that line of optimality. And this is actually a remarkable experiment when you think about it. I mean, here is a genome scale model that predicts the outcome of a pretty complicated biological process. But it's just at the phenotypic level, just the top level. You can't predict, you know, which genes got mutated, for instance, but you predict the overall outcome. And the underlying assumption is, like we've talked about before, is that the regulatory network and the kinetic constants just adjust through a selection process to achieve that optimal state. All right, so what do these things look like? Here is an example of a replicate experiment for that glycerol evolution. Growth rates improve over time, not always identically, and sometimes you get an outlier like that curve in red. Here is another type of an evolution where you're trying to get E. coli to grow on a non-natural substrate. And this is 1,2-propane-diol, which is very close to glycerol, but has, you know, alcohol groups on all 3 carbons. So what is done here during the first segment is that you have that non-natural substrate in there, but you have glycerol in there too, very similar molecule. You start taking that down, 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 down. So the growth rates goes down until you hit some crisis point there, and then some few key mutations show up that enable the organism to learn how to grow on propane-diol as a substrate. So that's an example. So one thing worth mentioning here in terms of automation, it turns out when you do automation, the experiments are much more reproducible. Passage is always done the same way. You know, the machine monitors the growth rate. That's the passage. Exactly the same volume is being passed. When you do this manually, people have to be coming in maybe at odd hours, and they may not always come in exactly at the same time, and there may be some variability in the passage volume and so forth. So the automation does this very well. And what is interesting is that in the automation, if you look at that curve carefully, there are these plateaus. So growth rate goes up and plateaus. So there's a fixation of some mutations, then there's recompetition at that growth level, and a new wave of mutation comes in, and some win out, and then there's a third one in that case. And these gray shaded areas it's hard to see it at the bottom, but you see a pink curve that goes up and down, and that's an estimation of the change in the slope of the curve. So when it goes to 0, you're on a plateau. When it goes off 0, you're growing fast. It goes back to 0, and so forth. So you can actually automate what's been called a jump finder, how you jump from one plateau to the next. If you look carefully at the outcome of the manual evolutions, maybe there are plateaus in there, but they're hard to see. All right, so a lot of these sort of experiments have been done. Strategic deletion of key genes in metabolism have been performed. And you make the deletion strains, and then you evolve each one of those deletion strains on multiple different substrates. So now the stringency in testing of a genome scale model and its prediction goes up a couple of notches. You remove a gene, and then you have to force the cell to economize its metabolism through selection without that gene. And you can do it on multiple substrates, so multiple inputs into that knockout strain. And the summary of those experiments are shown here. I believe we mentioned this before, but this is a summary figure that shows the computed growth rate or predicted growth rate versus the measured one. And the blue point there, blue points in this graph indicate the growth rate of the deletion strain right after the gene deletion. And the pink points are the end points of the evolution. And you see many of them end up on that 45-degree line, resulting in an accurate prediction of the outcome of the experiment. Okay, so this opens up the question of the objective function. And you can do these experiments under different conditions, like I mentioned. And there has been a question about the wild type strains. Are they really optimal? If not, why are they away from that optimum? If you evolve towards optimum, do you get all the way to the optimum, and so forth? So there are probably multiple objectives that an organism has. If you evolve based on the growth rate, maybe that becomes the predominant component of the objective. This panel here to the left comes from Uwe Sauer's lab at the ETH, where they looked at 3 different criteria, optimization criteria. The z-axis is the length of the flux vector, which is a proxy for having the minimal proteome expressed. And then on the x and the y-axis, we have maximizing energy production or maximizing biomass. And you see the experimental points, the red ones and the blue ones, lie on a plane that is formed in that 3-dimensional space. And in some cases, the experiments were done under carbon limitations, and some cases not. And the points migrate around in that space. And those little panels to the right with these histograms showing also a vertical bar, which is in red or black in here, it shows the estimation of some of the internal fluxes and how far they are away from the edge of the solution space. And the strains used here locate themselves close to the edge, but not on it, but there's some distance away from the edge. Now, I think as we talked about before in the class, the me model actually calculates automatically what is on that z-axis. It expresses the minimum proteome or the most economical proteome to execute the growth state. But it also, as shown in the panel to the far right, can compute the optimal growth state under substrate limited conditions or proteome limited conditions. And when you plot the outcomes of the me model, it forms that dashed line in that plane that is indicated in green in that icon towards the right. So all the points that are on that surface in this paper from the ETH kind of fall on that line. So the me model actually simultaneously can account for the 3 criteria shown in the 3-dimensional image here on the left. But still, and we're not going to address it in here, but there's still a question about how well evolved are strains, how close do they get to that theoretical optimum. Okay, so that's actually, that distance is subject to, I should say, experimental inquiry right now. All right, so let's talk about how we determine the genetic basis. So prior to 2007, it was very expensive to sequence genomes, even bacterial genomes. They would cost hundreds of thousands of dollars to sequence. Now, as I think we mentioned before, you can sequence them overnight for maybe $50 or so a genome. So you can take an outcome of an experiment from an ale machine that may have 50 endpoints and sequence them over a couple of days, and not for that much money. And you can even do population sequencing of the intermediate stages and use the read depth as a proxy for the allelic frequency, so you can see how the mutations are penetrating into the population as they're being selected for. Okay, so let's talk then a little bit about the genetic basis. So we have, of course, sequencers like that one shown in blue, and the accumulating experience with resequencing bacterial genomes after they evolve shows that all kind of genetic changes are observed. There are a lot of point mutations or SNPs that are observed, but you also have indels, which are insertions and deletions, and small-scale indels are surprisingly common, and sometimes there are multiples of 3, meaning that a discrete number of amino acids is added or deleted from a protein if they are occurring in the open reading frame. Sometimes indels can be bigger. When you're evolving eukaryotic genomes, you may see much bigger indels than that. So you can also see large-scale amplifications in bacteria, very often in between the rRNA operands. You may see that whole region duplicate because the rRNA genes are identical, so homologous recombination can happen between those. And those are maybe 100 kbs or more. Okay, so I should say that these mutations can lead to loss of function or gain of function. We may not talk about many cases. But the point is that when you do these adaptive laboratory evolutions, you see quite a bit of diversity and changes in the genetic material, and you can now define that. And you can repeat the experiments to see if the same mutations come up again and again, or whether the same genes tend to be mutated over and over again. Okay, so how do we determine the genetic basis? So this is an image that gives you an overall picture of how that is done. So on the top left, we have a fitness improvement experiment, growth rate, and you can collect cells along the way at different points in time. That kind of becomes like a fossil record for you. You just put it into the freezer and you can keep it there. You can streak out colonies at that point and freeze individual clones or just the whole population. And then when your experiments is done, you can resequence the genome. And you can find the genetic alterations. So they are indicated there in yellow, red, and a bluish color. And what you can then do, once you have these mutations defined, you can go into your fossil record and look for the allelic frequency of that mutation as a function of evolutionary time. And then you can get trajectories like shown in the top right, where the mutation isn't found in the beginning, it's 0. Then all of a sudden it appears and it gets selected for, and very often it drives to 100%. Because if you're doing this by passaging in the mid to late exponential growth phase, any mutation that confers a tiny growth advantage exponentially dilutes the competition out, and they tend to fix to 100%. Not always the case. Then what you can do is you can use the genome editing methods to introduce these mutations into the wild type strain, one at a time or combinations thereof. This has gotten a lot easier in recent years, where you can generate a large collection of multiple mutations and combinations thereof, and then screen that population for the best combination. Or you could just systematically put them in one after another, and reconstruct step by step all the mutations you find in the end strain. That is impractical for much more than 4 or 5 genetic changes, or at least was a few years ago. Maybe that has changed a little bit. But then what you can do, if you had these genetic changes introduced, you can look at the performance of the strain with those mutations in it and compare it to the wild type. And also compare it to the endpoint to see how much a mutation or set of mutations moves you from the wild type performance to the endpoint performance. And so on that graph, there is a bar from 0 to 100%, so 0 being the wild type performance and 100% being the performance of the evolved strain. So like is indicated in there, the bottom graph, you put the 3 mutations that have been found, and you may get fully recoup the phenotype of the endpoint, and therefore establish causality between the mutations and the phenotype that you observe. And then finally, the last panel on the bottom right shows that you may take the mutated gene products, clone them and express them, and try to figure out how their function changed. And that turns out to be a very difficult process and a very tedious process. Okay, so let me show you some actual data along these lines. So here are allelic frequency curves for mutations for the glycerol evolution. And if you look at the first panel in the top left corner, you see 2 mutations come in, fix very quickly, and reach 100% abundance. And these are mutations in glycerol kinase and in the RNA polymerase. Right below, you see 2 mutations fixed early on, which are also in glycerol kinase and the RNA polymerase. And you see a little graph, little curve that comes up and goes down. That is the same glycerol kinase mutation as is in the panel above. But here it didn't win out. It appeared and then disappeared. And some other mutation in glycerol kinase won out. And in that case, it's interesting. There's a 3rd mutation that fixes quite a bit later. And it turns out to be a causal mutation. But there's a very small effect on the growth rate. Or sometimes when they fix late like that, they can have a small effect on the growth rate. Okay, so that's how you can watch the history of that evolution and see how the mutations compete and how they win out over time. Okay, so the knock-in procedure is very simple. So here is one of the endpoints illustrated called GD, strain D on glycerol evolution, and it has 3 mutations in it, shown by blue, green, and red. And you can take the E. coli strain and introduce them separately, one at a time. And then you can grow those strains with just that single mutation in it and compare it to the wild type. And you may get something like this, where you don't, so the scale at the bottom doesn't go, well, the bars never reach 100%, but the scale goes to 100%. And here you see that if you put in mutations in the RNA polymerase, you get these red bars where maybe 40, 50, 60% of the growth rate change is recouped by only putting in 1 mutation. The blue bars correspond to mutations in the glycerol kinase in this case. And then the green ones are other mutations, and you see some of them towards the bottom do nothing to the wild type, but they're still selected for. And it turns out when you add them on top of the previous mutations, you can see their selection advantage. You can also take this process a step further. You can just put them in serially, like shown in that red box. You put them 1, 2, and 3, and now you have the wild type, 1 mutation, 2 mutations, and 3 mutations. You can compare them all at the same time. So you can reconstruct basically fully what you think the genetic basis is underlying the adaptation. If you think that the missing mutation there, which is the green one in the last one, is a hitchhiker, then it shouldn't have any effect, and the strain with the 2 and 3 mutations should behave the same way. So when you do that, you may fully reconstitute the growth rate of the endpoint. So that happens with the glycerol evolution, where 2 or 3 mutations come up, and when you put them in, the strain grows fully as the evolved strain. The reconstructed strain and the endpoint strain grow the same way. Another evolution shown here is on lactate, where there were a lot of different mutations. And in that case, it was very hard to reconstruct the endpoint because there were so many mutations to put in there. So in this case, presumably there are many mutations, as many as 10 of them, where each one of them has some contribution to the phenotype. But at the time these experiments were done, it was not feasible to put all 10 of them in. It's probably possible today with today's genome editing methods. So a simple genetic basis for the glycerol evolution with few highly causal mutations. For the lactate evolution, many, many of them, each one of them just adding incrementally to the fitness. Now it's interesting that when you're trying to assess causality here, you can actually use your mutations as markers. So let's say you wanted to compare the strain with the 2 mutations and the 3 mutations. And when you do the experiments just by themselves, you may not see a statistically different growth rate in them because the growth rates are very close. The mutation may only give a 2 or 3 minute change in the doubling time, and that's kind of hard to measure. But what you can do is you can just compete them against one another. You can just mix them 50-50, say. So yellow and blue as shown in the graph there. So yellow and blue, when you mix it, becomes green. Then you can observe the genetic composition over time. And if the blue strain wins out, it wipes out the yellow strain. Of course, this has to happen over a time scale where no new mutations can really fix. So this is the way you can get at the very fine effects of mutations on selection. And this is an example shown where I believe there was 2 strains being competed against one another. Well, in this case, 2 endpoints were competed against one another. One had 2 mutations, the other had 3. 2 mutations in the same gene product, and one had an extra mutation. So the curve there shows the allelic frequency of the 3 mutations, and you can see that they are basically disappearing here. So GA wipes out GE, I guess, in this experiment. So you get ratio the fraction of the loser to the winner, and you can estimate the differential growth rate between the 2, or sometimes you can score growth rates that are pretty small between the 2. So that's kind of fun to score them, to see what's good and what's bad, especially when you have multiple endpoints. You may have many, many endpoints and compete them all against one another. And I guess now with next-gen sequencing, you can just sequence the whole population and just look at the allelic frequency straight from the whole mixture. Now, then on to looking at what actually changes inside cells. So we know there are mutations, we can put them in, assess their causality, but what is the mechanism by which they change the function of the cell to make it more fit? Well, the first thing you like to do is to clone the mutated gene products and see how they compare to the unmutated ones and see how they differ. Now, it turns out this is a very, very hard process. What's illustrated in this slide is an assessment in vitro of the mutations in the RNA polymerase. So there's an in vitro assay on a fixed template, fixed DNA template, where you can measure basically the binding of the RNA polymerase and see how long it stays on the promoter, and then it goes off and starts making the transcript, and you can measure the elongation rate. And so down at the bottom are these 3D diagrams of the structure of the polymerase. You see furthest to the left the DNA fitting into the major groove there, and at the other end you see it's been rotated. You can see that through that groove, and some of the mutations are in that region where the polymerase grabs onto the DNA. And in the in vitro assay, you measure one substantial difference between the mutated form and the wild type form of the polymerase, and that is the half-life of the polymerase on the on the promoter, the so-called pause time, or the half-life of the time that the polymerase stays on the promoter before it starts transcribing. And there is like a 14-fold difference in that parameter. The mutated form just doesn't stay long on the promoter, just immediately starts transcribing the gene. So you can measure something like that. This, of course, is impossible to measure in vitro, in vivo, I should say, and it may be differential from one gene to the next. So this is kind of hard to deal with, but at least you know some of the kinetic properties of the polymers are changed. So that's an inquiry at the molecular biology level. It turns out that it is a little easier to do this actually at the systems level. So what this little diagram here shows is differential expression profiling between the wild type and that single gene, that single mutation knock-in mutant. And there are 3 of them shown here. And as you see, a large fraction of the genes are expressed exactly the same in the mutants as compared to the wild type. But there's a group of genes there that is specifically differentially expressed as a function of that single mutation. And you can actually look at them, and the downregulated functions there include motility, virulence type functions, TCA cycle goes down a little bit, so does oxidative phosphorylation, and some transport processes go down that are related to multi-drug resistance. And when this is, okay, let me show more of them. So so you can look at the sigma factor activities. So sigma F clearly loses activity somehow, but that's a very specific sigma factor for flagella and its function. But sigma S, which is a much broader acting stress response sigma factor, in its regulon, there's a bunch of genes that are also downregulated, very specifically. So stress functions are downregulated. So you can do a motility assay on the wild type and the mutant, and you see one moves and the other one doesn't move. And then you can look at these processes that are likely to change the growth rate or the yield. We have now studied this in more detail, and it turns out a lot of the downregulated functions are something we call fear functions or hedging functions. These are functions that the cell is upregulating and may or may not need. But if it's in an variable environment, it's ready to react to that. But when it's evolved under constant conditions, it specializes more and more and more, and those cells that drop energy and mass expenditures on those functions win out. So one way of interpreting this is that the cells become very greedy under that one condition, and their fear goes down. They're not fearful anymore because they have been growing in a constant environment for such a long time. They don't have to worry about things that they may otherwise have. So this actually is a way to measure the distance from that optimum, being exactly at the predicted optimum and where a cell actually locates itself. So now you have a biological measurement of that distance. You can measure this a Euclidean distance if you want to, but it's more important to know what the biological difference is. But moreover, you can now take multiple strains of the same species and evolve them all and see how they differentially locate themselves away from that optimum. And maybe based on that, you can glean or you can gain some understanding of how they were selected for. Why are they slightly different? And what was in their background that made them do things slightly differently? So that's a very interesting and creative use of adaptive evolution. Okay, so to date, there are dozens, I don't know, maybe 100 now, papers published on adaptive laboratory evolution. We showed some examples of changing environmental parameters here, the nutrition, lactate, glycerol, those have been done. But there have been a lot of other stresses put on cells, temperature adaptation, UV light, and so forth. These have been done for several bacteria. Saccharomyces, also being a eukaryote, has undergone adaptive evolution. Very interesting paper in Science 2 or 3 months ago from Jens Nielsen's lab at Chalmers University, but he found one mutation that conferred temperature resistance on yeast. It was just one mutation in one of the enzymes involved in lipid synthesis. Yeah. So you can learn a lot from adaptive evolution, many different things. So let's talk a little bit about the interpretation of the outcomes. So this is a summary of basically what I've been saying. So you do an ALE experiment, you have a start point, you have a trajectory, you have an end point. It is very easy to look at the genetic changes now. It's very easy to sequence, it's cheap. You can find the mutations. You can do this over and over again. You can get good statistics on how probable it is that a particular gene product gets mutated during that evolution before you start looking at the gene product. You want to make sure it's not just a spurious mutation if you only did the experiment once. It is also very easy right now to do the genome editing to put these mutations back into your wild type strain to assess causality. So that's easy now. At the bottom of this table is the phenotype. That's also very easy. It's easy to grow cells. It's easy to look at the metabolic byproduct secretion. It's easy to look at motility. It's easy to find out if the multi-drug resistance pumps are active. It's easy to do the phenotypic characterization. You may have a battery of tests you can do relatively easily. Now, what I showed you with that one mutation in the RNA polymerase, it is relatively easy, or maybe not easy, but it's kind of moderately difficult to assess the systems level changes, because you can do the omics data type generation on the wild type strain and the endpoint strain and even in between. And now that we have good reconstructions, you can map that data onto a reconstruction and you can interpret the changes that are happening. So that's something we can't do. So you can't deploy systems biology here. And that's maybe ultimately the most informative level of doing this analysis, because the cell is economizing itself. It's not just one gene, it's economizing its balanced function to be more fit. Now, the very difficult step in this process is the molecular biology and figuring out the mechanisms. And that example with the RNA polymerase kind of illustrates that. How are you actually going to establish the causal relationship mechanistically from that mutation to the function of a whole cell? Even, of course, that's a very broad-acting enzyme because it changes the whole expression profile. But even for other mutations, like the one in glycerol kinase, where you would think would be kind of obvious, it is not that easy to figure out how that mutation actually works. Okay, so genetic level, phenotypic level, easy, systems biology level, moderately hard, but molecular biology is very difficult. And in fact, I should just state that these two mutations in the RNA polymerase and the glycerol kinase took almost a whole thesis of students studying biochemistry. I mean, it took years and years to try to figure it out, and the answer wasn't complete. All right. Okay, so let's talk about some. There's a typo there. So, can we mimic natural evolutions in the lab and do experiments to try to understand what happens in nature? Evolving E. coli and glycerol probably doesn't represent a natural situation. It's an interesting situation, but maybe not a natural one. Okay, so let's talk about that a little bit. So I think we talked about the life cycle of E. coli before. The E. coli life cycle is characterized by shocks, certain shifts in environment. So for the first shift for an E. coli that's eaten by an animal is the temperature shock. It sits at all of a sudden it's at 37 degrees and it thinks maybe something's going to happen. Then it gets swallowed, and then there's a pH shock. When it's had those two shocks in a row, it's pretty sure what's going to happen next. Next, it goes into a fully anaerobic environment where there are a lot of nutrients and competing species. Then, of course, there's the fourth shock, which is the cold shock, where there's plenty of oxygen, things get cold, the nutrients evaporate away. So these are the shocks it has to be able to go through. Now, in the lab, you can actually implement these shocks and see how it anticipates changes. And for instance, when you do the aerobic to anaerobic shift, you see that E. coli just naturally opens up all the transporters for amino acids because it expects to go into a nutritionally rich environment. It anticipates that based on its evolutionary history. It just opens that up and expects the amino acids to be available. And of course, when you put them aerobically in glucose minimal media, it's very disappointed. There are no amino acids there. It has to synthesize them anyway. So you can do experiments like this in the lab. You can shift conditions over and over again and go through them repeatedly and see an adaptation. And this is actually from a paper where they did a heat shock, and then I think they inverted either the pH shock or the oxygen shock to try to get E. coli to take one shock and then react oppositely to the next shock than it does naturally. So you can do things like this. Here is something that would be very hard to mimic in the lab. So this is the process of salmonella infection. You know, it gets swallowed, it knows the temperature, pH, now it's in the stomach. Now it has to attach to the lining of the stomach, it has to, you know, cross the epithelial barrier, it may get engulfed by a macrophage, so it has to figure out how not to be lysed, you know, and so forth. And all along that path, it has pathogenicity islands. They're called spy islands, salmonella pathogenicity islands. And each one of them is upregulated under a different condition when it has to do a different thing to survive through this process. Now, I can't imagine how you would do an evolution in the lab to get through all these difficult steps. So I don't know how you, if you wanted to do an evolution here, you would probably have to do it somehow in an animal. I don't know what the readout would be, but this is an interesting process that is fully evolved and hard, I think, to tinker with experimentally. Here's another one that's a little easier potentially. So evolution to resistance to antibiotics can be done in the lab, and the mutations determined. It, of course, is not fully representative of what a cell may experience in the body because it interacts with other microbes. You know, it's not going to be a constant dose of antibiotics there and so forth, but you can at least identify some of the mutations that are likely to occur as pathogen acquires resistance to an antibiotic. So that's another type of experiments you can do. So in these three experiments I just mentioned, the objective function is very different than growth. There are many other things, serious things, that they have to be concerned with. Of course, they have to grow, but that's maybe not what they're optimizing. They're optimizing their chances of success, ultimate success. Okay, so at the end of the chapter, I have a couple of examples of complex adaptation. So this is a very interesting paper. This is colonization of a sterile animal. So there are sterile animal facilities around where mice are being bred or other animals in a sterile environment. So there's no microbiome in them. And very expensive, by the way, as you might imagine, to house these animals. And then you can feed them a defined dose of microbes. I think it's called a gavage. Sounds like a very fancy word, but it's basically force feeding, and you have to feed them, you know, exactly the same amount. And in this case, shows colonization by a strain of E. coli was put into these animals. And you can see this asymmetric evolution, which eventually stabilizes with 3 different subspecies. So 3 different strains survived the colonization, and somehow they learned how to coexist in the animal. So it's not like one strain takes over here, the community kind of forms. Another complicated adaptation is a long-term infection like Pseudomonas aeruginosa. In mostly males that have muscular dystrophy. Pseudomonas is an opportunistic pathogen. It lives in our respiratory tract. We all have our own strain of Pseudomonas that's adjusted to us. But because of the problem, I think, with the celiac hair or something like that in the lungs of those that have muscular dystrophy, the microbiome in the lung goes out of control, and over time basically kills the patient. And if I remember correctly, most patients with muscular dystrophy die in their 20s, if I remember correctly. But you can sample the lung microbiome over time, and you can sequence the Pseudomonas strain to see how it adapts to the host after a long-term battle with the immune system. And what's shown on the bottom there is a timeline, and this is in years, I think actually decades, where the green line shows the number of mutations that is accumulating over time. The blue line is the fitness function. So the first few mutations increase the fitness a lot, and then every subsequent mutation makes the organism less fit, but still more fit. So it's evolving in the patient. And there was just a paper published out of Søren Molin's lab in Denmark in Nature Genetics, I think last month or the month before, where 750 sequence Pseudomonas strains were analyzed that had been found in this patient cohort. So we can study, in other words, a long-term pathogenic process in this case. At the end, I just want to spend a few minutes talking about these causal mutations that you can think about as a new perturbation variable. You push exactly on the sequence there with a very particular change in the DNA sequence, and the organism changes. If you move over a few amino acids, you push there, maybe nothing happens. So these are very specific, highly sensitive spots in the genome of the organism. So, for instance, the RNA polymerase mutation that I mentioned, you push on the sequence there, and a few genes are differentially expressed, and nothing else. And now, for some of the point mutations that have been observed in the RNA polymerase, different amino acid substitutions in that position have been implemented, and almost none of them has the same effect as the wild-type mutation. I think there's only one other amino substitution that's found to have that causation. The other ones don't. So these are very specific perturbation variables, and you more or less have to find them by an evolutionary process. So to the top right is genome map of E. coli, and this is from a recent review, well, recent, now 3 or 4 years ago, that summarized all the mutations found by adaptive evolution in E. coli at that point in time from many different labs, many different stresses. And you can see they're kind of, at least at that point in time, distributed all over the genome. All sorts of gene products get mutated. Maybe as this data accumulates, maybe there are parts of the genome that tend to mutate more than others, but that's, of course, going to be biased by the selection pressure you're putting on the organism. One can maybe think about these perturbations in a three-dimensional space as indicated down there, where there may be physical, chemical, or biological changes in the environment, and the organism sits in some state before that change, the origin of that coordinate system, but it has to move away from it to another location, and that may be a function of only 2 or 3 of these very specific variables, these very specific mutations. And we can see these in the automated Ale machine, where you see these plateaus form. And if you start thinking about that abstract distance from the optimum in that space, you can maybe now scale it in terms of those perturbation variables and the associated processes with them. So exciting. Okay, so a summary. So the objective functions are being investigated here. You can use them to calculate optimal performance, and you can use them to predict the outcome of Ale experiments. The vast majority of those experiments to date have been constant environment. One environmental variable is changed. Some experiments are showing up where you flip-flop the variables in the environment in a time sequence, so it'll be probably quite a different outcome from that. So these evolutions are done in a controlled environment, and now cheap resequencing is possible. And what is also nice about bacteria is that the time scale of the experiments is short. You get a lot of cells, and you can repeat the experiment over and over again. That hasn't stopped people from doing these sorts of experiments with more complex animals. And I may have mentioned this before, but Gabby Haddad, who is chairman here of pediatrics at UCSD, have been evolving Drosophila flies to lower and lower oxygen tension. And I think he has colonies now growing at 4% oxygen or so. He resequences them now and looking at the expression profiling and so forth. So you can't do this for other organisms, but bacteria are easy for the reasons mentioned. Now you can use genome editing to take the mutations that you find, put them in the starting strain, and assess causality. You can have, like I said, the fossil record, if you will, and so you can look at the dynamics of the evolution, which is interesting because you can actually see a diversity of mutations come up on these plateaus, and then all of a sudden most of them lose out, but one of them drives it to the next plateau. So you can just watch the competition live almost. It's very tough to find out how a mutation changes the properties of a molecule, of an enzyme, DNA molecule, RNA molecule, and so forth. Because the mechanisms are just complicated. But mapping omics data onto a reconstructed network is helpful and helps you understand how a cell economizes its state. And more adaptive evolutions are being done now in creative ways, and a broad set of questions is being addressed. Let me just give you one example here at the end. We've talked a little bit about fluxomics as a data type, where you put a C13 isotope into your substrate and try to trace its fate going through a metabolic network so you can watch where the isotope goes. People have always questioned those experiments because the C13 bond is stronger than the C12 bond, and therefore the metabolic map that you're looking at has to be biased. Now, you can actually evolve E. coli on fully labeled C13 labeled glucose, and you can evolve it on C12, and you can compare the endpoints. And it turns out they're essentially the same. You can take the endpoints and flip-flop the substrates, and you get the same result. So that's maybe kind of it's not a scientific discovery, but it's at least satisfying to know that the label is not biasing the performance of the cell. Another set of adaptive evolutions that's being done now in the industrial biotech space is to see how the production strains can be tolerized to the product being made. And most of the industrial chemicals tend to be toxic, and if you want to produce them, 100, 150 grams per liter, so 10-15% weight fraction, the cells have to be pretty tolerant. The wild type strains will die immediately if you put them into those solutions, but you can tolerize them over time, and you can do it in an ale machine. And so forth and so on. There are just so many questions you can address using adaptive laboratory evolution. So that is the end of the lecture.